Hello, I'm Juan Jose Monterrey Martinez, and I'm going to be covering 4.2 prokaryotic cells. When I took a microbiology class, there was a lab that involved me putting my hand on a petri dish, putting it in an incubator, and then coming back a couple days and then observing that what was growing under on that petri dish under a microscope. And when I looked down the microscope, I was looking at prokaryotes. Those were just small single-celled organisms that have a relatively simple structure. What I could not see under the microscope were some of the other features that prokaryotes and eukaryotes share. So what I'm referring to is the plasma membrane, which is just a barrier between what is inside the cell and what is outside the cell. You could call it the cell wall as well. The cytoplasm is going to be all of the gelatinous sort of structure that holds all of the organelles that are also within the cell. And then the DNA is a little bit different in prokaryotes to eukaryotes because when you think of DNA, maybe you're thinking of a double helix. But when it comes to the prokaryotes, it's not. It's just a jumbled mess that's kind of generally in the center, but it is not in the double helix form, more of a circular. And additionally, there is going to be ribosomes inside prokaryotes that are going to be creating proteins that will be aiding in creating the cell wall or aiding in building any other organelles that are within the prokaryote. But the main thing here in the division where prokaryotes and eukaryotes divide is that there is no central nucleus compared to a eukaryote. Prokaryotes are just kind of generally tossed, the DNA genetic material is just generally in the center. There is no encased cellular membrane that keeps all the genetic material inside. So the unique thing that I observed in that laboratory where I put my hand on it is that not only do I have bacteria on my hands, and not only are we surrounded by bacteria in our environment, but there's also bacteria within us. So you have E. coli inside of your gut, and it's necessary and helpful because it aids in digestion. You also have different types of bacteria that's inside your body, and there's about an even amount of prokaryotes to eukaryotes inside of your body. Now, I'm going to home in on a bacteria that's called Staphylococcus aureus, and this bacteria grows and stays inside of your nose. Okay, let's do a little quick check-in. What are we looking at here? This is Staphylococcus aureus. Is this a prokaryote or is this a eukaryote? Well, what we're looking at here is an illustration on the left of what it would look like inside the cell, and then we have an electron microscope image of Staphylococcus aureus on the right. So we see it's pretty simple single-celled organism. Now, the reason that you're seeing that it's purple is because Staphylococcus aureus is gram-positive, which means when you dye it and then you put it under a microscope, it changes its colors into purple, so you can contrast it against the background. So on the left, we see the illustration shows that it's a very easy cell wall that's around here, and then we see our cytoplasm is kind of holding all of our ribosomes, and Staphylococcus aureus has a couple other things that we'll get to, but these things are called cytotoxins. So that's all inside there, but more importantly, we have no cell membrane enclosed nucleus. So there's no genetic material that is encased. The genetic material here is, like I said, just generally floating around the center. So it meets our criteria of a prokaryote because single-celled, simple, and that there's no cell membrane genetic material encased. Okay, so like I mentioned before, these single-celled organisms, Staphylococcus aureus, prokaryotes, are all around our body. The dangerous part is when they leave an area that they should be and then they travel to areas where they shouldn't be. Now, you can create this opportunity by, let's say, touching something that's contaminated, and then you touch your face, and then that you touch let's specifically your nose, right? Like I said, this usually lives in your nose. Staphylococcus aureus is responsible for a lot of respiratory infections, but it also lives on your skin. So it's also responsible for a lot of staph infections on your skin. If it gets into your bloodstream, that's when it gets really dangerous, and that it could become a situation where you become septic. So your whole body is now not just an even prokaryote to eukaryote, it's actually now more prokaryotes, and specifically that bacteria. So your body is overloaded with bacteria, and that can lead to a lot of health concerns. So we're talking about abscesses, right? And those are kind of like those nasty smelling pus, localized infections. It can lead to pulmonary diseases, and it can also lead to something that's called osteomyelitis, which is your bone has an infection, and sometimes that leads to surgery. So main point is that bacteria is within you. It travels all around, but it also, if it extends out to areas where it shouldn't be, then it poses a big problem. Okay, let's give a little context here. Put yourself in the position of Eugene Rowe. He is an actual person that's being portrayed here by an actor in the show Band of Brothers, but he was the medic for Easy Company, and he had a weapon that was really more effective than a rifle, and that was antibiotics. When World War II occurred, the mass production of antibiotics really ramped up to take care of soldiers. But in the future, or later on, better yet, antibiotics became overconsumed, and that caused what we are dealing with now are superbugs, where the bacteria has evolved and is now resistant to the antibiotics. So the way that antibiotics work is kind of like a lock and key mechanism, in which there's a lock inside the outside of the cell wall, and then the antibiotic comes in with that key, and once it enters, it's able to kind of disrupt a lot of the important functions of that cell. So it's the protein synthesis, or maybe it's the genetic synthesis. Oh, and once that is disrupted, the cell just starts to break down, and then you have more and more cells dying, and then you kind of get to that homeostasis level between prokaryotes and eukaryotes. But the thing is that because they're so simple, they're able to mutate very quickly and very effectively, in which is that what makes bacteria become resistant to antibiotics, is because they're able to mutate, and now they change their protein, and what used to be able to get in is now unable to. So the medication is ineffective because it's unable to enter that very simple cell wall, but now it doesn't match. So they have to create new antibiotics or different kinds of antibiotics in order for the medication to work. Okay, thank you very much. Have a great day.